Welcome to the second half of the carbohydrate lecture. I'm back from the safari and I have a special guest. Well, actually a few special guests have poured into my office again, as usual. Interesting. Okay. Thank you, Bart. That's really nice. Who else is here? Awesome! I love Krusty. Thanks for coming, Krusty. Wow, this is going to be some kind of lecture capture today.
Okey-dokey.
Yes. Alright, back to business. Types of polysaccharides. You need to know these. We talked about the monomers already. Now you need to know how they're put together. You also need to know that plants store their sugar as starch. Okay? That's how they store their short-term energy source. It's not the chemical energy of the cell. The chemical energy of the cell is always ATP. But short-term energy storage is starch. Plants. Animals do not store their sugar as starch. They store it as glycogen, usually in our muscle cells. We will see pictures of these, and you will need to be able to recognize these and draw cartoons of them as well. What's cellulose? Cellulose is the polymer found in the plant cell wall. Yes, cell walls are carbohydrates. Beta glucose. What's starch? Alpha. What's glycogen? Alpha. What's cellulose? Beta. Also known as what? Yep, you guessed it, people. It's known as fiber. We all love fiber, right?
Where's your sense of humor, man?
Exactly. What else? Chitin. What's chitin? It's the exoskeleton in the fungus, some algae, and many animals. What kind of animals? Spiders, arachnids, lobster. Yummy. Right? The outer shell, lobster, shrimp, beetles. That's chitin. Okay. Again, a different kind of monomer. Glucose with a little extra. Beta. Peptidoglycan. What the heck is that? Bacterial cell walls. Don't confuse cellulose with bacterial cell walls. Backbone of alternating monosaccharides, which again are a little bit different. Again, in beta. So, let's recap. Plants and animals, alpha, starch and glycogen, and cellulose is beta, chitin is beta, peptidoglycan, beta. Here we have starch. It's what? Alpha. Yes, alpha. Why? Why is it alpha? Because it just is. What is important about alpha? 1,4-glycosidic? We have enzymes that can break this. So we, as animals in our gut, can digest starch, send it through glycolysis for energy. We can break those bonds. Guess what we can't break? Beta. Why is fiber bulk forming for the feces? Because it's a beta-glycosidic linkage that we can't digest, so it runs through undigested. Fascinating. Alright, let's get back to this guy. Starch. Alpha 1,4. Thumbs up. Thumbs up. Both up or both down. Right? Generally, a bunch of glucose is linked together. Some are branched, some are unbranched. They call it a somewhat branched structure used for storage in plants. What kind of storage? Energy storage. The branches are 1,6. So if we looked at this, this would be attached at the 1 and the 6. About every 30. So somewhat branched. Some can be completely unbranched in starch. So you could think of low branching. Starch, alpha 1,4, plant. Glycogen, alpha 1,4, yes, because we can break it down to give us energy. This is what our muscles do. right? We use glycogen to generate ATP through glycolysis. It's highly branched. Now, the only way I'd ask you to determine which is starch and which is glycogen is if I showed you two pictures and whichever one had more highly branched would be glycogen. Otherwise, they're exactly the same thing. Alpha 1,4 glycosidic linkage, right? Thumbs up, or thumbs up, thumbs down. Storage of what? Energy. Branches again. 1,6 glycosidic linkages. So about every 10. So highly branched glycogen, animals, us, starch, plants, somewhat branched, lower branching. Alpha 1,4 is the important thing. We have enzymes that can break that. Now I have to say, cellulose is my favorite carbohydrate. Not only is it the most abundant polymer on this planet, but it's my favorite because it's fiber. Who doesn't love fiber? Exactly. Makes poop. Fiber makes good poops. Yay! Let's look at the linkages. A beta 1,4 glycosidic linkage. Right here, folks. Right? One up, one down. Can't be alpha. Alpha's both up or both down. Beta, up, down. Interestingly enough, it's not a single chain. If we look at glycogen and starch, yes, they're branched, but they're single chain, long chains. There's one branch at 1,6. One thing I didn't say, which I need to say, I made an assumption and I shouldn't have. Beta 1,4 glycosidic linkages, this guy, or alpha 1,4 glycosidic linkages are covalent bonds. Just like the other linkages, they're covalent. Peptide bond, phosphodiester bond, glycosidic linkage, covalent linkages. But what else do we see in here? What are these? What's this? It's hydrogens and oxygens, electronegative, electropositive. Holy crap. We're talking about fiber. Yes, hydrogen bonds. So if we want to zoom in, which of course we do, whoa, we would look at these guys are the beta 1,4 glycosidic linkage, glycosidic, covalent, yes, I like what you've done here, What about these suckers? Hydrogen bonds. So the individual covalently linked polymers are hydrogen bonded to another beta 1,4 glycosidic linked polymer, hydrogen bonded to another, hydrogen bonded to another, hydrogen bonded to another. Again, when I talked about covalent linkages, very, very, very strong. Hydrogen, not as strong. But when there's lots and lots and lots of them, they are strong collectively. That's what makes cellulose such a great polymer, such a great structural polymer. Right? Cellulose is what's in cell walls of plants. It helps hold them up. Cellulose is the wood of trees. Structural. Very regular repeating structure, very strong. Again, it's the beta 1,4, hydrogen bonding between parallel strands. We do not have any enzymes that can break this beta 1,4 linkage. So what happens? It just goes right on through. Now let's talk about chitin, the second most abundant polymer out there.
Boring.
Okay, come on, Homer. It's not that bad.
Boring.
Alright, we're doing it anyways, regardless of what he thinks. Chitin. Again, we're still talking carbs. It's a sugar, polysaccharide. It's a beta 1,4 glycosidic linkage. We can't digest it. if we eat bugs, munching on a cockroach would be just act like fiber. Go all the way through. It would help your digestion and provide bulk for your feces. Thank you, Homer. I really appreciate it. There's an addition onto the beta glucose, which is right here. An N-acetylglucosamine is what it ends up being called, right? This whole thing, instead of just glucose. And notice it has nitrogen, carbon, and methane group, sorry. So how, now suddenly it's going to be confusing. Again, all by itself, easy. But what if we now put this in with the amino acids? Holy crap, what do we see here? An NCC. Don't freak out. This addition is just like an amino acid stuck on there. It's an NCC. It's an amino acid. But it's attached to a glucose molecule in beta 1,4. So if I show you a picture that you need to identify, you will always get these two together, showing this beta 1,4 glycosidic linkage. I'm not going to leave this part in, so you can't just read that. Ugh. But you will see this whole thing. You'll know it's not an amino acid with an R group. Where would the R group be sticking off if it were an amino acid? Let's go back here. NCC, the R is always off the middle. Is the R off the middle? No, so it can't be an R. It's not an R, it's a sugar. This is a disaccharide that helps make up the chitin molecule. So again, chitin, 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 beta 1,4, beta 1,4, beta 1,4, beta 1,4, covalent, held together by hydrogen bonds. Okay, and then interaction here with the amino acid group, the N-acetylglucosamine that goes in between. Okay, again, very strong because of the massive amounts of hydrogen bonding and that sheet-like structure. The adjacent monomers are flipped upside down, right, so that these two, right, the other one, if we zoom in really quick, right, each one of these guys has, right, that's the additional NCC group here and the additional NCC group there so that they can interact with each other. Rather than them all being in the same direction, each strand is flipped upside down, okay? That's not how it is in cellulose. Cellulose, they're all the same side up. One up, one down, so it really doesn't matter. What about peptidoglycan? Again, oops, this is spelled wrong. Peptidoglycan. This is a polysaccharide in the cell walls of bacteria. Again, it's a beta 1,4. What does that mean? We can't digest it. What does that help us with? You betcha. And again, it's another permutation of the glucose, right? Thumb up, thumb down, beta 1,4 glycosidic covalent linkage. It's got the NCC stuck to it, so just like the chitin, looks very similar, but what else? It actually has a string of amino acids, alanine, glutamine, something I don't remember what it's called, alanine. And when it's set up, again, string of beta 1,4, beta 1,4, beta 1,4, beta 1,4, beta 1,4, beta 1,4, blah, 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 linked together. In this case, no hydrogen bonds. We don't see any here. No hydrogen bonds. Why? Why are there no hydrogen bonds? Do you see any place for hydrogen bonds? Well, yeah, you could really have this guy or this guy, but essentially the way they're linked is that these guys interact and this one interacts, and we actually can get a peptide bond between them. What? A peptide bond forms with the peptides or the amino acids on the other strand. Let's zoom in, shall we? So what are we saying? These bonds are the beta 1,4, covalent. This bond is a peptide bond. What is a peptide bond? Yeah, it's exactly like linking amino acids together. These green guys here are that string of amino acids coming off of this guy's string of amino acids coming off here, and we get an actual peptide bond there. And yes, it's covalent, so extremely strong. Parallel strands joined together by peptide bonds, covalent bonding. Super, super strong. So bacterial cell walls are extremely strong, very difficult to break down. We cannot digest them. So again, it makes up fiber. Okay, enough, Homer, we get it. So if you eat a whole bunch of bacteria, you're going to get lots of fiber. So let's all go into lab and drink some E. coli. Yummy! Fun facts about peptidoglycans include that most antibiotics are targeted against cell wall formation. So it's usually something that inhibits enzymes that build cell walls. So that means the cell walls are weak, they can break open, and they can be killed by antibiotics. Drug resistance occurs when enzymes evolve to not be stopped by antibiotics. So here's some more fun facts I've already been talking about, right? These guys are all together. Long strands bond between adjacent strands. Hydrogen bonds in the amino acids. Peptide bonds, right? Fibers are layered in sheets. That's why we call it fiber, because it looks like it's fibrous. Think of a celery. That's cellulose. Very fibrous. Try to chew that stuff up. Beta-1,4 glycosidic linkages. We can't break down, cannot hydrolyze, cannot break those. Alpha-1,4 glycosidic linkages. Yeah, we have enzymes. We use these for energy. We cannot use this for energy. No energy. We can't break those down. Very few can accommodate this. Okay? In our gut, only our bacteria break down any fiber for us. Okay? That's the only place it happens. Not our cells, not our enzymes, only the bacteria that live in our digestive tract. So they have lots of functions. Woohoo! We already talked about fibrous and structural. We talked about storing chemical energy, short term. What about indicating cell identity? What diverse functions in cells. Carbohydrates provide lots of structural support, right? This fibrous, water is usually excluded, blah, blah, blah. Beta-1,4, very strong, yada, yada, yada. We already talked about that. Cell identity, we haven't yet talked about. Glycoproteins, okay, or carbohydrates attached to proteins by covalent bonds are found on the surface of our cells, and that's how our immune system recognizes self versus non-self. The biggest glycoprotein we can think of are the blood group antigens, right? So type A sugar, type B sugar, that's all glycoproteins. So those are extremely important. Glycoproteins, cell recognition, right, and cell signaling, which we'll talk more about later in the semester and also ad nauseum in Biology 380. So here's a picture of a glycoprotein on the surface. This is the protein, an integral membrane protein that sticks in the inside. In here must be hydrophobic. Out here must be hydrophilic because there's all kinds of water out here. There's all kinds of water out here. Phospholipid membrane is hydrophobic. Storing chemical energy, right? We know we store glycogen in our muscles and some in our liver and some other places, and plants store energy as starch. We're going to talk more about that. Well, I don't even know what chapter it is in this book, but when we talk about glycolysis, okay, and energy transformation and usefulness in the cells. This is what we've already talked about, right? Alpha-1,4 we can break down. Beta 1,4 we can't. When we stick them through glycolysis, we get ATP. Okay. If you're asked questions, you need to know which ones store and which ones are structure. Glycogen and starch, storage, or all the alphas. Strength, mechanical strength, structure, all the betas. Right? Chitin, cellulose, and peptido. Don't forget peptidoglycan. Right? In the name it says peptide. Has to be the one with peptides with amino acids linked in it. And that's it. I'm glad you guys came, and I will see you soon.
Oh, Smithers, I guess there's nothing left but to kiss my sorry butt goodbye.
May I, sir?
How rude. Thanks for listening.